Good morning. Thank you, Horatio, and thank you to the organizers of the International AIDS Society for, in a plenary like this, having somebody talking about what I think are going to be sort of future medicines based on cell and gene therapies. It's a tough act to follow Gloria's talk, and especially I'm kind of aware of the irony of following a talk about resource-limited settings, talking about these types of therapies, but hopefully you'll bear with me. Okay, so to set the scene, I want to remind you of kind of what we're up against with HIV, the virus. It's a virus that hides from our immune system. It can mutate away from our best efforts at an immune response, and it can disable our immune responses. And some of these characteristics represent a formidable barrier to a vaccine. In addition, on top of that, HIV can also integrate itself into the genomes of our cells, and in some cases it can become transcriptionally silent and latent. And this latent form of HIV persists despite antiretroviral therapy, and it's the major reason that HIV is so difficult to cure. And of course, as you all know, it also is the reason that when people stop taking antiretroviral therapy, there is typically a rapid rebound of virus in the blood. So what can gene and stem cell therapies bring to this challenge? Well, I think of it in these three different categories. First of all, we can use genetic engineering technologies to think about engineering cells so that we can make uninfected cells resistant to infection. In addition, we can look at the immune responses that HIV so successfully overcomes, and we can think about ways that we can improve on nature and engineer our immune cells to be better soldiers going after this virus. And then finally, there are even technologies that we are developing now that could actually disable the latent reservoir that persists in individuals. So I'm going to be talking in particular about one type of gene therapy, which is called genome editing. And I recognize that many of you in the audience might not know what that is. So bear with me. I'm going to have a few slides that introduce the technology to you. So let's start with a simple definition. For me, genome editing is the ability to make precise changes in a cell's genetic information. And in order to make these precise changes, we need tools that can take the whole of the human genome that's packaged inside a cell, and they can seek out and identify the specific gene or DNA sequence that we are interested in. And some of the tools that have been developed that have that capability of recognizing specific DNA sequences are shown on this slide. They include the CRISPR-Cas9 system you may have heard about. CRISPR uses an RNA that is complementary to the DNA sequence of interest to bind to that DNA and bring in the associated Cas9 protein. Another way of doing this is to use protein-based systems such as those called zinc finger nucleases or ZFNs. And here there are proteins that are engineered to fit with the DNA sequence you want to target. And they also bring in associated proteins, in this case nucleases. And both CRISPR-Cas and zinc finger nucleases, when they bind to the DNA, they are able to cut the DNA and introduce a DNA break that marks that gene. Now, when you have a DNA break in your cells, and this happens to all of us every day in billions of cells in our body, the cell has very efficient repair systems that rush to break that DNA. And in human cells, the most common repair system is a process called NHEJ. And NHEJ sort of sticks the ends of the DNA break back together again, but it does this in a fairly imprecise way, and so you get little deletions or duplications of the DNA sequence at the break site. It effectively produces scars at the repair site, and these scars basically mess up the genetic code of your gene, and they effectively knock out the gene. So DNA break repair by NHEJ can be used to knock out a gene, and I'll be talking to you about how this has been leveraged to disrupt the CCR5 gene in a moment. in addition, because an integrated HIV genome is basically to the cell just another piece of DNA, you can also think about using these tools to target and disable an integrated HIV, a latent HIV genome. And indeed, these approaches are being sort of developed by a number of groups. I will draw your attention to a paper that came out very recently in Nature Communication showing in a mouse model that combining ART with these CRISPR-based nucleases targeting at latent HIV can actually lead to lack of HIV rebound in some of the animals. So knocking out a gene, quite honestly, is not that useful most of the time. You know, our genes do things, that's why we have them. So an alternative approach that gene engineers are interested in is, rather than using the NHEJ pathway, instead to hijack a repair pathway called homology-directed repair, or HDR. And HDR uses a repair template to copy the genetic information and thereby repair the DNA break. And what we can do is we can hijack that process, because we can introduce into the cell a repair template that fools the system. And our repair template can include modifications that we might want to make to that gene, thereby allowing us to mutate a gene. Or we can even put in a whole extra piece of DNA, and we can have that inserted in a very precise site-specific manner at the site of the DNA break. So those are the sort of two main technologies I wanted to introduce you to. And on this table here, I'm summarizing the different ways that different scientists are thinking about applying these to our challenge of suppressing and curing HIV. In the interest of time in my presentation, I'm just going to focus on two specific examples. I'm first of all going to tell you about a gene knockout example with CCR5, which is now in clinical trials. And then I'm also going to tell you about some initial data from my lab doing site-specific insertions. Before I move on to that, the final piece of background information I want to touch on is the idea of engineering stem cells, and in particular the hematopoietic stem cells, or HSC. HSC are the stem cells that live in our bone marrow and that produce all of our blood and immune system. And so this means that if you engineer a stem cell, you can have an impact on a multitude of different cells in the body, including the cells that HIV infects, such as CD4 T cells, and the cells that come to try and fight HIV are CD8 T cells, or natural killer cells, and our antibody-producing B cells. And all of these are available if we go into the hematopoietic stem cell to engineer it. And a final important point is that by engineering a stem cell, we hope that we can develop therapies that become a one-time treatment, because a hallmark of a stem cell is it lives forever and it produces its progeny forever, so that by engineering a stem cell, you may have a lifelong therapy. Okay, so the first example I'm going to talk about is how gene editing and gene disruption was used against the CCR5 gene. And it's probably not surprising to you to understand why this was the first ever target for genome engineering. CCR5, as you know, is the major co-receptor for HIV, but importantly, about 1% of the world's population can live without the CCR5 gene, and this is because they carry two copies of a defective, or if you like, a knocked out version of the gene called the CCR5 delta 32 allele. And what's been sort of absolutely igniting the field was the observation that patients undergoing bone marrow transplants, or HSC transplants, because they have a blood cancer, and receiving their transplant from a CCR5 delta 32 donor have been shown to be cured of HIV. The patients who are in this category include the Berlin patient, who is now more than 10 years out from his bone marrow transplantation, is not on ART apart from as PrEP, and has no HIV. And in addition, at Croy earlier this year, 2 new patients were reported, albeit they are at much shorter periods of being observed, the so-called London patients and the Dusseldorf patients, who are funded by a consortium called ICSTEM. So what we want to do as genome editors is say, can we recapitulate these observations from bone marrow transplantations from Delta 32 donors? And the idea here is that rather than having a transplant from a donor, you instead have your own stem cells removed from your body, engineered to make them CCR5 negative using the tools I've told you about, and then returned into the body. So if you like, it's a self-transplant. And there are currently several clinical trials that are ongoing to test this idea. ICSTEM are continuing to identify and recruit patients who need to undergo stem cell transplantation for an underlying blood cancer and trying to match them up with CCR5 Delta 32 donors, so to keep repeating, if you like, the Berlin patient observation. In addition, there are 2 sets of trials using both T cells as well as hematopoietic stem cells that have been engineered with zinc finger nucleases. These have been run out of UPenn and City of Hope in Los Angeles, and the City of Hope trial is the one I'm going to be giving you an update on. And then finally, in kind of like a combination therapy, Beijing University is taking patients who need to have a stem cell transplantation but don't have a Delta 32 donor, and they are instead editing the stem cells of the donor to try and recapitulate the Berlin patient observation. So I'm going to be giving you an update on this trial here. So first of all, just as background, my involvement with this was almost 10 years ago now. In my lab, we took some of these gene editing tools, the zinc finger nucleases, and we showed that if we edited them at human hematopoietic stem cells with these tools, and then took these stem cells and put them into a special strain of mouse, that mouse, in a matter of weeks, basically grows as a human immune system, and we can then infect the mouse with HIV. These are called humanized mice. And what we showed is that if we look at the levels of HIV in the mice, for mice receiving untreated normal human stem cells, they got infected with HIV and the HIV levels stayed high. In contrast, the mice that received the CCR5 zinc finger nuclease treated human cells, although they initially got infected by HIV, they were able to control the infection and it disappeared below our limits of detection. And this was because over time, the CCR5 negative cells that were present in that edited stem cell infusion became the sort of dominant cells in the mouse and were resistant to HIV, and HIV had nowhere to go. So taking this initial proof of principle, we formed a team of scientists, clinicians, and my group based in California, and we were able to receive funding from the California Institute of Regenerative Medicine, which funds stem cell research in California. And this is just showing you the timeline of how we went from this sort of preclinical observation to continue with the necessary preclinical development and the sort of regulatory development that eventually it ended up in 2016 with our first patients being enrolled in a clinical trial. This is the more detailed information about the clinical trial, showing you its official title, the clinical PI and the other investigators at City of Hope and the biotech partner Sangamo Therapeutics. And importantly, the sort of clinicians out in the community who identify, enroll, and will follow up patients who are enrolled in this clinical trial. This is probably my busiest slide. There's no way to show this simpler. This is the schematic of how this clinical trial happens. In the first part, the stem cells from the patient are isolated from their body. Those stem cells are then engineered to make them CCR5 negative. That manufacturing process and checking and everything takes about 6 weeks. And then there's a period where the patients then come in and are given treatment with a drug called busulfan. There are 2 different cohorts with 2 different doses of the drug. And busulfan sort of prepares the patient to receive back their engineered stem cells. And then there'll be a follow-up period for up to 15 years because this is, after all, a gene therapy. And the other thing I want to point out to you is here that at the 9-month period, if individuals are shown to have CCR5 negative edited cells that can be detected in their blood, they may then undergo an analytical treatment interruption. They stop taking their antiretroviral drugs. And the reason that's done is because we want to see if the treatments had any effect, and also because even if HIV starts to come back, we sort of imagine that the edited cells will have a selective advantage and they will be selected for and may be able to control the virus. Importantly, with any type of ATI, there are eligibility criteria that have to be met before we would subject a patient to this treatment. And there are also very rigorous stopping rules to put the patient back on ART should the virus start to come back. So this is a summary of where that trial is, and I'm grateful to the clinicians and scientists for allowing me to present this data. 14 patients were enrolled, 8 have been treated, and 2 of those individuals have gone on long enough that they're currently at the ATI stage. The data in the slide is showing you the levels of CCR5 edited cells that can be detected in the blood now out to over 600 days for the first individuals who are treated. And you'll notice there are 2 colors, blue and orange, and the higher levels of CCR5 edited cells that can be detected are in the orange individuals. And these are people who are in the second cohort, so they received a higher dose of the drug that allows engraftment of the stem cells, and they also received higher doses of the zinc finger nucleases. And then ongoing and follow-up studies are very typical for these types of trials. There is assessment of CD4 subsets and functionality and HIV reservoir analysis. So I look forward to hearing more about these trials as more of the individuals reach the stage of the ATI. So I thought I would just pause at this point and reflect on something that people have now started asking me, which is, here you are 10 years after you started thinking about this, and is CCR5 still a good target to you? And there's a couple of things that, maybe give me some pause. One of them is, earlier this year, you may have heard of what's been called the CRISPR baby controversy. This was a case where a scientist in China announced that he had taken embryos, human embryos, from a couple where the father was HIV positive, the mother wasn't, and he had used CRISPR gene editing technology on the human embryos to knock out the CCR5 gene in those embryos. The embryos were reimplanted into the mother, and at least 2 baby girls were born. This was met with widespread condemnation, not least because there is a de facto moratorium on any embryonic gene editing. As a society, as a species, we haven't yet wrapped our head around whether we should or could be doing this. So certainly the investigators' actions were seen as those of a rogue scientist. For me, as somebody who works on gene editing and has been using CCR5 as a target, the fact that he also used the CCR5 gene in, I think, a rather cynical ploy to do this offends the heck out of me. I believe CCR5 was chosen because there is already some human experience of doing CCR5 editing, so that's used as a justification. But more so, I see the cynical reason that was given, that he wanted to help these potential children by editing these embryos because their father was HIV positive. You know as well as I do that there are far, far simpler ways to stop somebody becoming HIV positive than editing them as an embryo. In addition, the other thing that's been in the news lately that is related to CCR5 was this report that came out a month or so ago from a large study in Britain that identified for the first time that people who are naturally CCR5 negative, again, it's about 1% of the population, do actually have some consequence for that mutation. Because when you look at the likelihood of being alive at 76, you'll see that people who are homozygous for the delta 32 allele do have a lower rate of that probability. So this just reminds us all that as we are doing any type of intervention, including a gene editing, even if we believe that it will have a benefit, that we always have to think about the risk-benefit equation. And I will say at this stage, we don't know if this difference which occurs to people who are, 100% CCR5 negative in every cell in their body would also hold true for people where a fraction of their hematopoietic system is CCR5 negative, but it certainly bears thinking about. Okay. So in the last few minutes of my talk, I'm going to switch gears and instead give you some examples of what I think we can do to use this other pathway in gene editing, the HDR pathway, to do some precise site-specific insertions of anti-HIV genes. And thinking about the types of anti-HIV genes that I'd like to introduce, I'm a big fan of genes that get secreted and can be present throughout the body and therefore have the potential to give a benefit not just to the individual engineered cells, but to basically every potential target cell for HIV in the body. And the idea behind this is that we would engineer some cells in the body. These can be blood cells, but they could also be muscle cells, for example, and that they would secrete anti-HIV factors. And therefore, the engineered cell becomes a factory. And some of the factors that have been proposed to be secreted are things like broadly neutralizing antibodies that could block HIV entry. What genome editing brings to this scenario is because we have the ability to precisely insert that anti-HIV gene at our chosen locus in the genome, we can improve the safety of the procedure, and we can also choose a site that's very amenable to having high levels of expression of our factor that will persist for a long time. In addition, you can also think about having a kind of like a two-pronged approach and combining this with CCR5 disruption by actually inserting your gene at the CCR5 locus so you both knock out CCR5 and also express your, for example, broadly neutralizing antibody. And some of the studies we're doing in my group, and I'm going to show you just one slide about that, are using an inhibitor called ECD4IG, that we believe may be an even more potent and broader inhibitor than broadly neutralizing antibodies. So what is ECD4IG? So ECD4IG was developed by the lab of Michael Farson. He's at Scripps Jupiter. And it's basically a very potent entry decoy. It contains parts of CD4, but it also contains a peptide that binds to the co-receptor binding site on the HIV envelope. And so in this way, ECD4 should block HIV from entering a cell. In my group, we work with Michael to test out whether ECD4 as a recombinant protein could prevent HIV infection using the humanized mouse model I've already introduced you to. And we found that compared to mice that didn't receive ECD4, the mice receiving the protein were able to persist through 2 challenges with high-dose HIV, showing its potential. Because of that, we've gone on to now use ECD4 in genome editing approaches. And again, as I explained, we insert it into the CCR5 locus. This is just showing you for a group of mice that the edited cells we can detect in the blood of the mice persist over time, having between about 10 and 30% of the cells have at least one allele that expresses this factor. And not surprisingly, we then see levels of the protein in the serum that increase over time. And when we challenge the mice with 2 doses of HIV, and importantly, even though they should be protected against CCR5, in these experiments we are using an X4 tropic HIV. You'll see that compared to the control mice, we see a significant protection. So putting broadly neutralizing antibodies, or ECD4, into a locus like CCR5, I'm sure it will work. But if you think about it, it's a long way from the natural situation where, for example, an antibody is expressed from a locus called the immunoglobulin locus and is only expressed from cells such as B cells. So we're also interested in working on whether or not we can edit the actual immunoglobulin locus. And the rationale here is that if we express an antibody from this natural locus, not only will we get secretion of the antibody, but we can also get some other features that are the characteristics of antibodies, including the potential for ongoing evolution in the face of a virus. So in order to do that, which really is a very big challenge for genome editors, we've been working with Dennis Burton's group at the Scripps Institute, and this is some data from a paper we published earlier this year, and it's largely the work of James Voss, his postdoc. And James is using a sort of a double cutout and replace gene editing strategy to insert a broadly neutralizing antibody at the immunoglobulin locus. And excitingly, we can detect that gene editing works because the edited B cells can now recognize and bind HIV. So this is kind of the early stages of this project, but I think it's a very exciting potential direction to really start to engineer an immune response against HIV. So to summarize, I hope I've convinced you that genome editing can allow precise changes to be made to a cell's DNA. CCR5 knockout is already being evaluated in clinical trials, but there are going to be other applications, for example, inserting anti-HIV genes at specific locations, including secreted entry inhibitors. And in addition, genome editing has also can be used to target the actual integrated HIV genome and thereby disrupt the latent reservoir, although I didn't have time to talk about that today. And I'll just leave you with some final thoughts. Maybe you're already having some of those about whether or not this is actually right for HIV. I'm talking about complex gene and cell therapies for a disease that you can treat with drugs. And as we heard from Gloria's talk earlier, we're already having issues doing that. So how on earth could we roll this out in resource-limited settings? The safety of genome editing has not yet been established, although clinical trials are now starting. What about public acceptance of genome editing? Pricing, it's safe to say it's expensive as heck right now. How do we think about, bringing that price down? And I think one of the things I'd be looking for from the future is for the scientists working in this area to develop methods whereby we can do in vivo delivery to take these genome editing tools and inject them into a patient so that we can get editing in situ without this need for complex ex vivo cell manipulations. So I'll just leave you with acknowledging the current and past members of my lab, the CCR5 zinc finger nuclease editing team at City of Hope and Sangamo, and my collaborators working on the antibody and ECD4 editing. And of course, a huge thank you to the large group of different types of funders who step up and support this type of work. Thank you.